Which neglected tropical disease is not caused by viruses, fungi, or bacteria? The answer is venomous snake bites. Snakebite envenoming, classified as a neglected tropical disease by the World Health Organization, impacts millions of people every year, leading to thousands of deaths. Antivenoms are effective, but they come with health risks and production accessibility issues. To that end, while microbes may not be the cause of the snakebite problem, they are part of the solution. Scientists are coupling microbiology with biotechnology to create snakebite antidotes that overcome some of the limitations of antivenoms. And it goes both ways. That is, they're not just using microbes to help combat venom, they're also using venom to combat microbes. Welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM. Before we dive into today's video, please take a moment to fill out the survey linked in the description. It takes less than 3 minutes and will help us make better videos. We appreciate your support. Every year, an estimated 5.4 million people are bitten by snakes, and over half of these bites can lead to envenoming, or the injection of venom into the wound. The outcomes of snakebite envenoming can be dire, ranging from paralysis and tissue destruction to fatal hemorrhage. Of those envenomed by snakes, an estimated 138,000 people die, though the actual number may be much larger. Most snakebite envenoming occurs in low and middle income countries in Africa, Asia, and Latin America, where access to healthcare and medical resources is limited. Women, children, and farmers often suffer the brunt of the impact. In 2017, the World Health Organization designated snakebite envenoming as a neglected tropical disease and aims to halve the number of deaths and cases of disability caused by envenoming by 2030. But some experts think this is a lofty goal, with progress failing to track with intent. Nevertheless, treatments are a big part of advancing that progress. And currently, treatments for venomous snake bites come in the form of antivenoms, which consist of antibodies derived from animals like horses or sheep. To generate antivenoms, tiny volumes of venom are injected into the animal over several weeks. Antibodies, including those that target venom components, are extracted from their blood. When injected into a patient, the venom-specific antibodies bind and neutralize the venom. The key is to get a patient the necessary antivenom as soon as possible because, while it can prevent future damage from venom, it can't mitigate existing damage. Now, though effective when administered in a timely manner, antivenoms aren't perfect. Because they contain all the antibodies and proteins in the blood of an animal they were derived from, not just those targeting the venom, a patient's immune system may attack those proteins, leading to potentially deadly allergic reactions. Beyond the health risks, production of antivenom is costly and requires advanced quality control. Many regions with the greatest need for antivenoms also don't have the capacity to regulate or produce them, and accessibility is a chief concern. Moreover, antivenoms often have a limited capacity to cross-neutralize venom from different snake species, meaning a specific antivenom must be generated for each species. Now, with these limitations in mind, scientists are harnessing the powers of microbiology and biotechnology to create novel venom antidotes. Specifically, they are using phages, or viruses that infect bacteria, to help generate recombinant antibodies, with a particular focus on special antibodies sourced from animals like llamas, camels, and alpacas. Now, the reason these antibodies are special has to do with their structure. Whereas quote-unquote conventional antibodies consist of 2 pairs of heavy and light chains, these antibodies only have 2 heavy chains. Scientists can home in on the variable region at the top of those chains that recognize a protein of interest, such as toxins in venom, and use that small fragment as a therapeutic tool. This small fragment is called a nanobody. There are several reasons why nanobodies are attractive solutions for dealing with envenoming. For one, they have similar high affinities and specificities as standard antibodies, but are more stable at high temperatures and low pHs. Nanobodies also have an increased ability to penetrate deep tissues because of their small size, and health risks may be lower as nanobody formulation would not contain all the other proteins that may be present in standard antiserum. Now, the process of obtaining antibodies usually involves phages via what's known as phage display technology. We'll use a recent example in applied and environmental microbiology to explore how this works. In this study, researchers derived nanobodies from an alpaca immunized with venom against Naja atra, or the Chinese cobra, one of the most prevalent venomous snakes in China. To generate the nanobodies, the researchers first immunized an alpaca with N-venom over time and collected blood, just like what is done for antivenom production. The scientists then extracted RNA from immune cells in the blood of the animal and generated complementary DNA from those sequences. The resulting DNA sequences included those that encode these special heavy chain antibodies found in animals like alpacas. And using PCR, the scientists amplified the variable region of those sequences, that is, the part of the antibody that encodes a nanobody. That small DNA segment was then inserted into a circular DNA vector, which was then expressed in phages. When they infect bacteria, namely E. coli, the phages begin to replicate and express the nanobody on their surface, with different phages expressing different nanobodies. In this case, the phages were then isolated and applied to N-venom in a process known as biopanning. The idea behind biopanning is to see which nanobodies expressed by the phages bind best to the venom. Those are the ones that show the most promise. The sequences of the selected nanobodies can then be expressed in bacteria and purified on a larger scale. This capacity to produce nanobodies in bacteria could enhance cost-effectiveness and efficiency. In this study, the scientists homed in on 4 specific nanobodies, all of which showed high affinity for N-venom in vitro. Mice, subject to a lethal dose of venom, had an 11%, 22%, or 67% survival rate depending on the nanobody they received. This was less than what was observed with antivenom, though the scientists highlighted that the 67% survival rate is quote-unquote noteworthy. They mentioned that more work is needed to evaluate the nanobody's therapeutic efficacy, as what happens in mice doesn't always translate to humans. What the study does show is that phage display technology is a solid method for identifying nanobodies with high affinity for snake venom. Additionally, the scientists found that the nanobodies did not cross-react with 6 other widespread snake species commonly found in Southeast Asia. They did, however, show cross-reactivity for venom from other Naja species, indicating it could be of broader use for multiple species, not just one, as is common for antivenoms. To that end, mixing individual monoclonal nanobodies targeting different venom components could help boost their efficacy and species coverage. A study in Nature Communications used phage display technology to identify nanobodies from animals immunized with venoms from various North American coral snake species. The results showed that a mixture was just as good as antivenom at neutralizing the lethality of whole venoms from different coral snakes. The individual antibodies, however, were not effective. According to the authors, nanobody mixtures may have the potential to significantly reduce batch-to-batch variation, neutralize all medically relevant toxins in the targeted venoms, and ensure a high therapeutic antibody count in recombinant products. Ultimately, this is a stepping stone toward finding alternative ways to manage venomous snake bites. Now, while we're thinking about how microbiology can be employed to counter the effects of venom, it's also worth mentioning that the relationship is a two-way street. Venom can also be used to understand and combat microbes. In fact, venom from diverse organisms, from snakes to bees to spiders, harbor antimicrobial components that can be used and engineered for therapeutic purposes. In a recent example, venom from honeybees and the oriental wasp, a type of hornet, showed in vitro activity against SARS-CoV-2, the cause of COVID-19. Wasp venom, in particular, reduced viral load of SARS-CoV-2, as determined via detection of the virus nucleocapsid antigen, in nasopharyngeal samples from patients over time, hinting at its potential antiviral uses. Other examples have shown that bee venom can be an alternative to antibiotics in treating bacterial infections, like those caused by Staphylococcus aureus. Proteases in snake venom can inhibit biofilm production by bacteria and may be leveraged as an antivirulence molecule in the fight against antimicrobial-resistant infections. Venom itself also contains microbes. Exploring these venom-dwelling microbes can lend insights into how they adapt to extreme environments. Moreover, some may be pathogens that cause infection in humans. If they are transmitted via a snake bite, for instance, this may indicate that infection care is another aspect to consider when managing bites. All in all, this so-called venom microbiome is a ripe area for additional research. That's all for today. Don't forget to take the survey linked in the description. I want to thank you for listening, thank Ray Ortega for production, and I'll talk to you next time.